Okay, good afternoon, everybody. We're go ahead and get started in the interest of staying on time. So I'm Jay Ponder. I'm going to be the moderator for the 4-hour and 10-minute session this afternoon. So if I pass out about 5:30, somebody can prop me up. So our first speaker this afternoon is Greg Ross, who worked with John Chodera at Memorial Sloan Kettering, and more recently, I guess, is at Schrodinger. We've been having a discussion about whether it's Schrodinger or Schrodinger. We should probably decide that since everybody's using their software. So Greg's going to tell us about how to deal with salt conditions in realistic simulations.
Thanks very much, Jay. So, as he mentioned, this work was carried out with John Chodera at Memorial Sloan Kettering. I'm now at Schrodinger. So, I'm talking about salt. And when we look at salt in the body, we find that there's very different concentrations depending on where you look. So in the intercellular fluid, the salt concentration is dominated by sodium chloride. But when we look inside the cell, the potassium ions dominate and acids and proteins dominate the negative charges. And even inside organelles as well, there's a great variety of salt concentration. So in the nuclear plasm, there's way more electrolytes, so lots more sodium and lots more potassium, for instance. And malignancy also can change salt conditions. So tumors are well known to alter pH, but they also alter the ion concentrations. So in tumors, if you look at the extracellular fluid around tumors, there's an enrichment of potassium ions. And this is because necrosis causes cells to spew out their intracellular potassium to the extracellular fluid. And this has an effect that it actually suppresses the immune response. And this plot shows, it's from this paper in 2016, showing that in mouse and human tumors, the potassium concentrations are greatly elevated with respect to serum levels. It should go without saying, but ions also greatly influence biomolecular behavior. So in addition to ion channels, which are an obvious example, they are also known to affect things like protein folding and solubility, the affinity of ligand DNA complexes, the affinity of host-guest complexes, and the affinity of small molecules and proteins. But when we do fringe calculations or molecular dynamic simulations, we sometimes only do neutralizing counterions or some fixed amount of salt, typically sodium chloride. And the fact that it's fixed is interesting because when we look at the local environment of a protein or biomolecule, the number of ions within a local region is constantly fluctuating. So if you imagine that this test tube is some macroscopic system, it kind of is, within this test tube, the amount of salt is fixed, but in any local region around a protein, the number of ions is constantly increasing or decreasing. And it's fluctuating a lot. But we don't really know how much it's fluctuating by. Also, depending on the properties of the protein and how it interacts with ions, it may draw in more positive ions than one would expect based on the macroscopic salt concentration or fewer. So with this in mind, we have some motivating questions that we wanted to address with simulations. First off, what's the mean salt concentration around a typical biomolecule? Secondly, what is the degree of fluctuation of salt around a biomolecule? And thirdly, to what extent are binding for energies dependent on the mean salt concentration? To what extent are they dependent on the variance of the salt concentration? And perhaps more practically, how much should we add when we're doing molecular dynamic simulations or fringe calculations. And it's probably worth saying that different simulation packages have a totally different way of dealing with salt. So what is the best, if we could know the answer, what's the best way to add salt? So we tackled this problem using semi-grand canonical Monte Carlo. Now, you could also tackle this problem by simulating explicitly the volume we're interested in and explicitly simulating the reservoir. So we could do molecular dynamic simulations of a huge system that exchanges ions between the reservoir and the system of interest. But in GCMC, Grand Canonical Monte Carlo, we take the limit of this reservoir becoming infinitely large so that the number of salt pairs and water molecules tends to infinity, but the ratio of them stays constant. And this mathematical trick allows us to replace explicitly simulating the saline reservoir in this case with just the system of interest, and the methodology allows us to create and annihilate ions within that. And I'll refer to any device that's able to maintain a constant macroscopic salt concentration, but a fluctuating variable amount of ions around a biomolecule and osmostat. So we wrote an open source package called SaltSwap, which is available on GitHub in the Chodera Lab GitHub account. And I use Young and Cheatham parameters, and we only consider sodium chloride for this package. We do have a paper out. It's in the bioarchive, and it's going to be shortly coming out in JPhys Chem B. So the way that the method works is by exchanging salt ions with water molecules. So it's called a semi-grand canonical Monte Carlo because the total number of molecules in the system is constant, such that when we insert and delete an ion, we're exchanging that with water. So total constant number of particles. And the fact that we have this variable number of water molecules means that we have to have a new simulation parameter, and that is this chemical potential difference, which is the difference between two water molecules and sodium chloride. And the reason why it's two water molecules is because we're exchanging two water molecules for every sodium chloride. And I should also say that all these transformations are electrostatically neutral. Now, the important thing to remember with the chemical potential, or the chemical potential difference here, is that there's a one-to-one mapping between the salt concentration in the reservoir, which we're not simulating, and the chemical potential that we're using in the simulation. So the chemical potential here is actually a property of the reservoir. And if we have a salt concentration in the reservoir that we'd like to reproduce in our simulation, we need to have the right delta mu. And a bit more mathematical detail, this is the probability density that we're sampling from in the SaltSwap package. So it's dependent on, say, the total energy of the system, which depends on the configuration X. We do have a barostat, so that's the pressure and volume. And you can see here, this is the extra semi-grand canonical Monte Carlo term. And I define the number of salt pairs in the system as the minimum between sodium and chloride. So basically, it's the number of neutralizing pairs in the system. Okay, so how do we insert and delete ions? So the problem in doing an instantaneous insertion and deletion is that it's probably going to be rejected. And so instead, we use non-equilibrium candidate Monte Carlo to gradually annihilate the changes to the system. So the way it works is that imagine we've got two water molecules, both with their non-bonded parameters, their partial charges and van der Waals parameters. Over a non-equilibrium trajectory, we gradually permute these to the non-bonded parameters of ions, such that in the end state, we have two ions. And in the process, we've allowed the system to relax around these changes. And the trial moves are accepted or rejected based on the non-equilibrium work of this process. And in our hands, we didn't try to optimize it too much, but we found that non-equilibrium candidate Monte Carlo really accelerated the sampling. So if you do instantaneous insertions and deletions, the probability to accept something is like 10 to the minus 50. But with NCMC, you can get sort of 30% or 15% depending on the water model. And this shows that as you increase the length of the non-equilibrium process, there's a monotonic relationship between the acceptance probability and the length of the protocol. And the reason why tip 4p seems a bit lower is I'm not sure, but there must be a lower phase space overlap with the ions in tip 4p. And if we look at the efficiency of the simulation as well, defined by, say, the average probability to accept over the total compute time, there's a massive boost in efficiency to something like to the 10 to the power 50. So NCMC is crucial to being able to sample salt concentrations. And I can't go into too much detail on how we calibrated the osmostat, but as I mentioned, there's a one-to-one relationship between the chemical potential and the concentration of the reservoir, and the mapping depends on the free energy. So this is the equation we use to map chemical potential to concentration in the reservoir, and you can see that it's dependent on, if you can, it's a big equation, but it's this F free energy here is the free energy to swap two water molecules, and this V is the average volume in the system. So to calibrate the chemical potential, we have to calculate these for a number of different salt pairs. The basic workflow goes like this. We do self-adjusted mixture sampling, and to calculate different salt concentrations for a range of different parameters, and we calculate the free energy to insert or delete salt based on the non-equilibrium work. And based on these, on the curve, the resultant curve, we can say, oh, I want to simulate at 200 millimolar, and you just read off the chemical potential you want, and then you've got yourself an osmostat when you apply it to simulation. So when applied to a protein, DHFR, you can see that the salt concentration fluctuates over time, and these are different macroscopic salt concentrations, and that the autocorrelation time is on the order of, say, a nanosecond. So if you want roughly tens of independent samples, you need to run for tens of nanoseconds. So returning to the motivating questions of how much salt do we expect to see, biomolecules, and what's the best scheme to add salt, we applied the osmostat on DHFR, Sark kinase, and a Drew Dickinson DNA dodecamer. And the plots I'm about to show define the local salt concentration as the number of salt pairs over the total volume. And current methods to add salt in fixed salt MD, you can either add salt, say, based on the total simulation volume, such as in GROMACS, the volume of the solvent, such as in CharmGUI, or the ratio of water molecules to salt pairs, such as in OpenMM. And this is always after neutralizing counterions have been added. So, without further ado, this is what realistic biomolecule salt fluctuations look like. In a box of water, we can see that the fluctuation is actually very large. So this is for a 200 millimolar osmostat. So the fluctuation goes from, say, 100 millimolar to 300 millimolar, and all schemes are quite good at reproducing the salt concentration. With DHFR, the only one that seems to do quite well is the ratio, based on the ratio, and again, the degree of fluctuation is very large. Sark kinase, the fluctuation is much shorter because the number of water molecules is larger. But with the DNA dodecamer, which has a total charge of minus 22, all methods do not capture the actual mean salt concentration very well, and the variance is much, much larger. And that's because there's an awful lot of neutralizing counterions in the DNA dodecamer system, and that has an effective salt concentration on its own, so that acts to repel other ions. And in the process of writing this up, Jeremy Smith got in contact with John and I and gave us, let us know about his paper that he released, and that had a mean field approach to predicting the local salt concentrations in biomolecular systems. And you can see that it does okay at all these systems, but does a much better job at reproducing the mean in the DNA system. So I can see I'm out of time. So I think I should wrap it up. And but that's the main conclusion, basically. So I don't have time to go into anything more, but I'd just like to thank those at MSK, particularly John Chodera and Bas, who helped a lot. And I would have talked a little bit about water and the sampling of water in GCMC and further extensions, but with that, I'd like to thank Lingla as well, particularly. So thank you.
Any questions for Greg?
I haven't actually. So at this, that's a great question. And at this stage, it's more like it's getting the infrastructure available to ask those kinds of questions. So I don't know, but I would love to see. And I'd also like to see what effect it really has on binding ranges as well. So that's really, those kinds of questions are the next steps.
Translate the 100 to 300 millimolar to actually the number of ions you're talking about in the context of the, I think, probably 25. I forgot how small it's possible.
Yeah, I can't tell you exactly, but it's a small number. So, I mean, no more, well, in a system containing maybe, say, 5,000 water molecules, it's going to be like 5 sodium chloride pairs, something like that. I think it's pretty small because it's millimolar. Yeah, it's a handful of ions.
Can you comment on the timescales involved in terms of equilibration of the ion atmosphere, especially with respect to what might be a charge-changing perturbation? What can folks sort of expect in terms of that?
Yeah, that's a really good question. So in terms of, yeah, okay, so there's two ways to answer that question. One way, a direct way, is saying, how long do I need to propagate the dynamics of the system such that they appear relaxed? And the NTMC, this non-equilibrium candidate in Monte Carlo, is kind of approaching that. So it's something like, so a high acceptance rate basically means there's good overlap between, that you've allowed the system to relax enough around, say, a new ion. And that's something like 20 femtoseconds. But that's probably too short. So that's the first answer. The second answer is specifically looking at the autocorrelation time of interactions around a biomolecule. So this is looking at different autocorrelation times for the DNA dodecamer. And the green, there's a lot of different lines here, but the green line here is the autocorrelation time for a fixed salt simulation. And so you can see that after around 3 nanoseconds, there's no autocorrelation. So, but with using the osmostat insertion and deletion, the autocorrelation time is much, much less. So I think it depends on precisely what time scale you're looking at. Does that address your question?
It's a start for sure.
Okay, good.
Okay, let's thank Greg.